Hello, my name is Nikhil Dereddy, and this is my project. How does the type of inhibitor affect enzyme activity? Purpose and background information. Purpose. I want to see what kind of inhibitor can most effectively slow down pepsin digestion. Competitive or non-competitive inhibitors. Because if I could, I would be able to see what kind of inhibitor is best suited to prevent stomach ulcers and other problems in the digestive system that involve excess pepsin activity. Background information. Pepsin digests polypeptides, long chains of amino acids, into amino acids, simple molecules that can combine to form proteins, and can cause ulcers if it digests the mucus that makes up the stomach lining. Biuret reagent is a liquid that turns purple when reacting with polypeptides, but not lone amino acids. With lone amino acids, it turns pink. Inhibitors lower enzyme activity, and there are two main types, competitive and non-competitive inhibitors. Competitive inhibitors compete with the substrate, material that the enzyme breaks, to bind to the active site of the enzyme, the area of the enzyme where the substrate binds to get broken down, which slows down the breakdown of substrate. But the effect of the competitive inhibitor could be reversed if the concentration of substrate is high enough. Non-competitive inhibitors bind to an allosteric site, the site of the enzyme that isn't where the substrate binds. This binding then changes the shape of the active site, not allowing the substrate to bind to the enzyme. Unlike competitive inhibitors, non-competitive inhibitors don't have to compete with the substrate to bind to the enzyme. Research question and hypothesis. Research question. Which type of enzyme inhibitor, competitive or non-competitive, best inhibits the enzyme pepsin's ability to digest proteins? Hypothesis. My hypothesis is that the non-competitive inhibitor will be the most effective at inhibiting pepsin's ability to break down proteins because they will have little competition when binding to the enzyme's allosteric site. But competitive inhibitors would have to compete with the enzyme substrate to bind to the active site. Material list. 30 ml of 1 to 3,000 activity pepsin, 5 large eggs worth of egg white, 500 ml of laboratory grade Biuret reagent solution, buffer capsules, pH 2 vial, 10 pipettes, 5 glass containers, 20 test tubes, 2 liters of distilled water, 1 stopwatch, 1 measuring cup, 1 paper towel roll, 100 grams of polyphenols extracted from green tea powder, 10 grams of peptide A, and 16 fluid ounces of 91% isopropyl alcohol. These are my procedures. 1. Gather all the materials needed. 2. Label a glass container for the pH 2's buffer solution. Dissolve 2 of the pH 2 buffer tablets in 200 ml of distilled water to make the pH 2 buffer solution. Then pour this into the glass container labeled pH 2. Then pour the large eggs worth of egg white into the unlabeled glass bowl. Then label 2 pipettes for the 2 inhibitors, competitive and non-competitive. Next, label 2 glass bowls for each inhibitor, competitive and non-competitive. Step 7. Mix 100 ml of isopropyl alcohol with 0.1 milligrams of peptide A, and then put in the glass bowl labeled competitive inhibitor. Step 8. Mix 100 ml of distilled water and 0.2 milligrams of 50% polyphenols from green tea extract, non-competitive inhibitor, in the glass bowl labeled non-competitive. Step 9. Label a glass bowl pepsin and add 30 ml of the pepsin and 90 ml of distilled water. Step 10. Prepare a row of 12 test tubes. Step 11. Prepare each of the test tubes by first adding 2 ml of the pH buffer solution to each of the test tubes. Then, add 1 ml of the competitive inhibitor solution, 1 ml of egg whites, and 1 ml of the pepsin solution to each of the test tubes. Then gently swirl each of the test tubes. At every 5-minute interval, add 1 ml of Biuret reagent in one of the test tubes till the Biuret reagent color goes from purple to another color, usually pink, and hasn't continued to change color in the next 5-minute intervals. Record your observations. Step 15. Thoroughly wash the test tubes. Step 16. Repeat steps 10 to 15 with 1 ml of non-competitive inhibitor solution and also without inhibitors. Step 17. Repeat steps 10 to 16 10 times in total for 10 trials. Step 18. Make conclusions based on your observations. Results and data analysis. The title of my data table is which type of inhibitor inhibits pepsin the best. All units are in minutes. The average for the control is 17 minutes. The average for control in green tea is 44.5 minutes. And the average in minutes for peptidin A is 40 minutes. The results and data analysis for this is labeled here. And this is my other table. Not table, I mean graph. And these are the photos. And this is my data analysis. The averages in my experiment for the 3 different groups, control green tea, which is the non-competitive inhibitor, and peptidin A, the competitive inhibitor, are 17 minutes, 44.5 minutes, and 40 minutes, respectively. This shows that the control group took the least amount of time, showing that the inhibitors work. It also shows that the non-competitive inhibitor works better than the competitive inhibitor at inhibiting protein digestion because the pepsin in the non-competitive inhibitor solution took longer to digest than the pepsin in the competitive inhibitor solution. The results happen this way because of the way these inhibitors work. Competitive inhibitors have to compete with the substrate to bind to the enzyme, so it's hard for the competitive inhibitor to inhibit the enzyme. Since non-competitive inhibitors don't need to compete with the substrate to bind to the enzyme, it is easier for the non-competitive inhibitor to inhibit the enzyme. My experiment supports the fact that non-competitive inhibitors are better able than competitive inhibitors at inhibiting pepsin from digesting protein. My results support my hypothesis that the non-competitive inhibitor will be the most effective at inhibiting pepsin's ability to break down proteins because they will have little competition when binding to the enzyme's allosteric site. But competitive inhibitors would have to compete with the enzyme substrate to bind to the active site. This is shown in the data when it says that the non-competitive inhibitor made the pepsin take longer to digest on average than the competitive inhibitor. My method may not be consistently reliable. This is because the accuracy of the time I've recorded for my tests is only in 5-minute periods. Also, since my experiment's results are based on the color of the solution, I may have inaccurately determined when the protein was digested. A strength of my experiment is I didn't have to buy large amounts of materials because the intervals in which I recorded the time were 5 minutes. Also, there are most likely some errors in my data since I have a few outliers in each dataset. My procedures could be improved if I use smaller intervals in between the time I have to measure and if I use an objective app to measure the color of the solution. Conclusion. The results of this experiment support my hypothesis that the non-competitive inhibitor will be the most effective at inhibiting pepsin's ability to break down proteins because they will have little competition when binding to the enzyme's allosteric site. But competitive inhibitors would have to compete with the enzyme substrate to bind to the active site. The result of my project is that non-competitive inhibitors are better than competitive inhibitors at inhibiting pepsin from digesting proteins. My project is connected to the real world because pepsin is used by your stomach to digest proteins. My project also benefits society because it can be necessary to try and slow down pepsin's protein digestion if it is causing problems like stomach ulcers, which can be caused if pepsin digests the protein in the stomach's protective mucus lining. Future recommendations and applications. Some future recommendations for this experiment would be to shorten the intervals in which I measured how much protein was left in the solution, used easier to get inhibitors, and used a more objective means of finding out the color of the solution after adding the biorite reagent. Further study I would recommend for this experiment is to try different types of non-competitive inhibitors and see which one works best. Some further study that could be tried is to see how the amount of substrate affects the ability of competitive and non-competitive inhibitors to inhibit pepsin. Some next questions to ask would be if the inhibitors I use, peptidin A in green tea, are able to inhibit some other enzymes that are similar to pepsin. Acknowledgements. My mom and my dad, Miss Manning and Miss Cramp all helped me through this science fair project. And these are my references in my bibliography. So these are the sources I use to research.